Now we are going to study first category of amino acids, that are amino acids with non-polar side chain. Now, what are these amino acids? These amino acids have non-polar side chain that does not gain or lose the electrons, and they even don't participate in hydrogen or ionic bonding. So in these cases, in case of these amino acids, the R group is actually a oily group or a lipid soluble group. It promotes hydrophobic interactions instead of hydrogen bonding or the ionic bonding. Now, what are the locations of these R groups in case of these amino acids? For example, in proteins found in aqueous solutions, a polar environment, the side chain of these amino acids, the side chain of these non-polar amino acids tends to cluster in the interior of the protein. So this diagram is actually representing that we have a non-polar amino acid that is represented by this yellow color, and we have a surrounding environment that is aqueous solution. So what will happen in that case, your R group of the amino acid will cluster in the interior of the protein. So, but if we have a polar amino acid that is represented by the red color, it will cluster on the surface of the soluble protein. So this is difference between the non-polar amino acids and the polar amino acids locations of the R groups. Now, this is like the formation of oil droplets in the water. Similarly, just like the formation of oil droplets in the water, if we add oil in water, you can see that there are droplets in the water. So similarly, in this case, if we have a non-polar amino acid and you are introducing it into a aqueous solution, you are introducing it to a polar environment. What will happen in that case? Your amino acid will tend to cluster in the interior of the protein. So what will happen? This will make a three-dimensional structure. So by filling up the interior of the folded protein, it is actually making up a three-dimensional structure. So these non-polar R groups are actually making the three-dimensional structure. So what will happen that if these proteins are located in the hydrophobic environment, such as in the cell membrane. Now these non-polar R groups will tend to cluster on outside surface of the protein. That is, they will interact with this lipid bilayer. So in this case, the non-polar amino acids, which are represented by the yellow color, will cluster on the surface of the membrane protein, and thus they will interact with the lipid bilayer. Now, again, this figure is representing the nature of the amino acids and nature of the environment in which they are introduced. This figure is representing that the yellow color circles are actually representing non-polar amino acids. In case of the aqueous solution, they will cluster on the interior of the protein, thus making a three-dimensional structure. While in the case of the lipid soluble environment, non-aqueous environment, these amino acids will cluster on the surface of the protein, thus interacting with the cell membrane. While on the other hand, if you have polar amino acids that are represented by these are rectangular boxes, they will cluster on the surface of the soluble protein. So this was all about the amino acids which were having non-polar side chain.